Hello, everyone, and thanks for coming. One fast announcement before I hand it over to Liz to introduce our first speaker, and that is that the monthly Myco talks are next on April 28th, and it'll be two great speakers, Kyla Ost and Gustavo Goldman, who will talk about Candida species in the gut and Aspergillus fumigatus. So I'll put the registration info in the chat. Liz?
Hi, everyone. Thanks so much for joining us. I know we're competing with Nilesh Govinder's professorial talk today, so thanks for popping into this seminar series. We've got two fantastic speakers. Our first speaker today is Spyridoula, who's going to talk to us about candidalysin. She received her bachelor's degree in biology from the University of Athens and then mastered in immunology at King's College London. She stayed in London and got her PhD from the Crick there, and then recently started a position at UCL. And I think she's going to tell us about the work that she has done at UCL and at the Crick, probably. Take it away, Spyridoula.
Thank you for the intro. So, see my screen? Okay. So, as you may know, an increase of invasive fungal infections has emerged worldwide. Poor diagnosis accompanied by ineffective drug therapy and lack of a prophylactic vaccine hinder the elimination of these infections. In healthy individuals, Candida albicans resides as a harmless component of their mucosal microbiota. However, a dysregulation of the immune system or even a breach in the epithelial surface caused by trauma perturbs the balance between host and fungus, leading to diseases which contribute significantly to morbidity and, in more severe cases, mortality in millions of people every year. It's very important, especially for immunocompromised patients where antifungal immune responses are impaired. The main site that this fungus must breach in order to become pathogenic is the mucosal epithelial surface. The cells on this surface can discriminate between different fungal morphologies via induction of a biphasic MAP kinase immune response. Candidalysin was discovered as the missing link between cell damage, between hypha formation and cell damage. It can also induce epithelial immune activation. So this schematic represents what we know from candidalysin so far. Candidalysin intercalates into the invasion pocket created by a Candida hyphae and intercalates the host cell membrane, inducing lactate dehydrogenase release, which is a cell damage marker, and calcium influx. The latter activates matrix metalloproteinases, which shed EGFR ligands, which in turn bind to EGFR and activate downstream pathways and the c-Fos transcription factor, which results in the release of many pro-inflammatory cytokines and chemokines. Given that in the Candida albicans infection model, we know that c-Fos is activated predominantly downstream of P38, we also presumed that candidalysin activates an EGFR P38 c-Fos pathway, although this was never experimentally confirmed. Moreover, we know from previous data that candidalysin induces EGFR internalization via a mechanism which is poorly understood. Therefore, the aim of my PhD was to delineate the activation mechanisms of the MAP kinases and how are these orchestrated along with EGFR to promote inflammatory cytokine release. To start with, determine the role of MAP kinases in cytokine induction by candidalysin and how are these activated by EGFR. To approach this, we used the TR146 cell squamous carcinoma line, which presents high ultrastructural similarity with the human buccal epithelium. For our experiments, we treated these cells with inhibitors or siRNAs and then assessed pathway activity. As a main readout, we assessed cytokine induction. So to give you a sense of how most of my experiments looked like, this is a typical fluorescent Western blot of the TR146 cells pretreated with this MAP kinase and EGFR inhibitors prior to candidalysin stimulation. In every slide at the bottom corner, I will show you the inhibitors used each time and their respective targets. This is a Western blot developed with the LICOR system, which allows accurate quantification as it does not permit signal saturation. And after quantification and data analysis, we saw that candidalysin activates an EGFR ERK c-Fos pathway, while P38 appears to sit outside of this axis. So next, we assessed our readouts, which is the inflammatory cytokine release of these 5 cytokines and chemokines. This heat map represents cytokine release in cells treated with these inhibitors compared to candidalysin alone stimulated cells. The lighter the color, the stronger the inhibition of each readout. I will focus firstly on interleukin-6, as this was the only cytokine found to be regulated only by P38 among the MAP kinases. So therefore, it served as a useful biomarker of pathway activity. Inhibition of P38 had no impact on the transcription levels of IL-6, suggesting a post-transcriptional control of this cytokine by P38. Indeed, inhibition of P38 reduced IL-6 levels in both cell supernatants and cell extracts. And when we checked for the involvement of the eukaryotic elongation factor 4E, which plays a major role in elongation of the translation, we saw that P38 inhibition significantly reduced its phosphorylation at 2 hours post-candidalysin stimulation. Therefore, P38 controls IL-6 release at the translational level. Next, we focused on GM-CSF, as this was shown to be regulated by all 3 MAP kinases, but in particular from ERK downstream of EGFR. From previous data of the lab, we know that GM-CSF is activated via c-Fos downstream of EGFR. But since P38 does not sit in this axis, we sought to investigate how could P38 regulate GM-CSF release. For this reason, we focused on P38 known substrate, the heat shock protein 27. This is an anti-apoptotic peptide which interferes with the apoptoson to block the actions of procaspase 9. Knockdown of HSP27 reduced GM-CSF release. And the convolution of this pool sRNA showed that and confirmed the independence of the P38 signaling pathway from ERK. So now that we know that candidalysin induces 2 distinct pathways, we sought to investigate the triggers of these pathways following candidalysin stimulation. Treatment of cells with cetuximab, which is an EGFR ligand binding neutralizing antibody, so it blocks binding of the ligands on EGFR, reduced phosphorylation of ERK, as well as c-Fos activation, but did not totally abolish it, suggesting that EGFR ligand binding on the receptor is a trigger for the downstream pathway, but not an exclusive one. Then, during the course of these investigations, we noticed that P38 inhibition with 2 structurally different P38 inhibitors could inhibit phosphorylation of EGFR at 3 tyrosine residues, which are important in MAP kinase activation and EGFR dimerization, showing a role for P38 in mediating EGFR phosphorylation. This phosphorylation appeared to be independent of ligand release, as P38 inhibition did not impact release of epiregulin and epigen, 2 ligands known to be released quite early and at very high amounts post-candidalysin treatment. So therefore, we know now that P38 and EGFR ligand binding are 2 mechanisms inducing EGFR phosphorylation. And our next step was to assess the contribution of each mechanism. For this reason, cells were treated either with cetuximab alone or with cetuximab plus the P38 inhibitor. Cetuximab alone significantly reduced phosphorylation of all these 3 tyrosine residues. And even increasing concentrations of the inhibitor had no further impact on its phosphorylation, suggesting that any remaining phosphorylation is not attributed to ligand binding mechanisms. However, when cetuximab was combined with a p38 inhibitor, this phosphorylation was even further decreased, even below the baseline levels, confirming therefore that p38 and EGFR ligand binding are two distinct non-redundant mechanisms of EGFR phosphorylation. So why did we focus so much on the p38 regulated EGFR phosphorylation? As I've told you in the introduction, we saw previously that candidalysin could induce internalization of EGFR. So using live cell imaging and an EGFR antibody, which stains the cells red, as you can see here, first we confirmed that candidalysin does induce EGFR internalization. You can see the red dots here entering the cell, which likely appears to be a recycling mechanism, although they have not proven this yet. In the next video that starts now, cells are pre-treated with a p38 inhibitor before candidalysis, and you can see GFR remains accumulated on the plasma membrane with a minimal internalization that is similar to baseline levels. So therefore, candidalysin induces a p38-driven EGFR recycling. So p38 does not sit downstream of EGFR, but rather upstream. We still, though, ought to identify what activates p38 following candidalysin stimulation. The canonical mechanism of p38 activation involves the MAP kinases 3 and 6, which were found to be phosphorylated in the presence of candidalysin. Knockdown of both these MKKs reduced phosphorylation of p38 by approximately 50%, as well as its downstream substrates, HSP27, phosphorylation of EGFR, and IL-6 release. It is clear, though, that only a component of p38 activation occurs via upstream MAP kinases. So next, we sought to investigate additional non-canonical activation mechanisms. And we focused on the Src-family of kinases, since these have been shown in the literature to activate p38 either as upstream triggers of the MKK 3 and 6 or independently of them. We used a broad-spectrum SFK inhibitor called PP1, and we saw a reduction in the phosphorylation of p38, as well as its downstream substrates, EGFR phosphorylation, and HSP27. However, PP1 did not impact MKK 3 and 6 phosphorylation, further suggesting that in our system, SFKs contribute to p38 phosphorylation independently of the MKK 3 and 6. While PP1 and MKK knockdown individual treatments reduce phosphorylation of p38, when we combine these two treatments, phosphorylation is even further decreased, suggesting non-redundant regulation of p38 phosphorylation. But what was more interesting is that among these two treatments, PP1, meaning SFKs, were the main regulators of the EGFR phosphorylation via p38, while MKK3 and 6 appear to be the dominant regulators of IL-6 release via p38. So it seems that we have differential signaling outputs from p38 depending on its upstream trigger. So our next goal was to investigate how could this happen. We assessed phosphorylation of p38 on two different residues. First, the canonical dual residue, which is located in the activation loop, and the non-canonical residue called tyrosine 323, which has been shown to be phosphorylated in T cells via another member of the SFK family called LCK. And phosphorylation on this residue causes a conformational change in the structure of p38, allowing it to autophosphorylate its activation loop. In our system, while both SFKs and MKK3 and 6 appear to regulate phosphorylation of the dual residue, only Src appeared to regulate phosphorylation of tyrosine 3-3. It's worth mentioning here, I said Src because when we used another SFK inhibitor, which is Src selective, called dasatinib, it yielded the same pattern of responses with PP1, and therefore we presume that Src is the member of the family activating P38. So to summarize this part of my talk, I've shown you so far that candidalysin activates an EGFR ERK c-Fos pathway via EGFR ligand binding on the receptor. Of course, this is not the only trigger. And P38 is activated via distinct upstream regulators, which differentially control its signaling outputs. Src controls the P38-driven EGFR phosphorylation, while MKK3 and 6 appear to control IL-6 release downstream of P38. But both pathways contribute to HSP27 phosphorylation. Given the complexity on the P38 activation mechanism, we next sought to examine its role in an in vivo mouse model of oropharyngeal candidiasis. For this reason, female BALB/c immunocompetent mice were pretreated with a P38 inhibitor for every 12 hours, starting 1 day prior to infection and up to harvesting. On day 0, mice were anesthetized. A cotton swab soaked in yeast culture was placed sublingually for 75 minutes. And at days 1, 2, and 4, mice were euthanized, their tongues were excised and used to assess oral fungal burdens. At day 1 and 2, P38 inhibition significantly increased the oral fungal burdens, while at day 1, also mice presented a lower body weight. By day 4, mice fully cleared Candida albicans and their weights returned back to normal level. We also assessed whether this involvement of P38 in early fungal clearance would require the induction of innate type 17 response. So we checked for the transcription level of these known genes, but unfortunately we didn't see any impact following P38 inhibition, suggesting that the role of P38 in early fungal clearance is independent of raising type 17 innate immunity. Finally, to confirm all our in vitro work, we performed Western blot in the tongue lysates of these mice. And first, we confirmed that the P38 inhibitor reduces phosphorylation of P38 at both days 1 and 2, while c-Fos is not affected by P38 inhibition, which aligns with all our cell culture work showing independence of P38 from c-Fos. And our collaborators from Pittsburgh, from Sarah Gaffen's lab, who developed c-Fos knockout mouse, they also showed that knockout of c-Fos had no impact on early fungal clearance, as well as on type 17 immunity, which shows that c-Fos appears to be dispensable in acute candidiasis, which was really surprising for the field. So this is the summary of the schematic that I showed you before. In the future, we aim to confirm whether the P38-driven EGFR recycling is also downstream of Src or not, and whether this mechanism here could be the reason why we see an early fungal clearance with P38, which could probably involve its role in maintaining EGFR on the plasma membrane. And also we need to confirm this hypothesis we have on the compartmentalization of the signaling. So where is P38 activated, by which trigger, and whether any scaffolding proteins are involved. Hopefully, after answering all these questions, we will have identified more targets for future therapeutic interventions. Closing, I would like to thank my two supervisors, Peter Parker at the Crick and Julian Macklick at King's College London, and my two labs for making this PhD such a joyful experience. Especially big thanks goes to James and Natalia for performing the P38 inhibition in vivo work during lockdown. Our collaborators from Sarah Gaffen's lab, Bianca and Chunsek, for the c-Fos knockout work. And thank you all for coming here today.
Thanks, Spyridoula. It was a beautiful talk. I'm sure we'll have lots of questions. We're going to go directly into Josh's talk, so I'll just pass it back to Jessica to introduce him.
Great, thank you very much, Spyridoula. That was wonderful. And please enter your questions into the chat. We'll take them all at the end. So I'm thrilled today to introduce Josh Kerkaert. He's coming to us today from Geisel School of Medicine at Dartmouth College. But before that, he completed his bachelor's at the University of Minnesota Twin Cities, where he studied environmental biology of endemic fungal pathogens. And today he's going to tell us about his thesis work from his PhD in Rob Kramer's lab on Aspergillus fumigatus and biofilms.
Is this showing the right screen or is it flipped again?
Right now, we're still seeing the presenter view.
Okay, cool. There should be good.
Perfect.
Awesome. So yes, thank you everybody for coming. And today I'm going to talk to you a bit about the work that I've been doing with Rob Kramer and looking at this and these really interesting findings that we found regarding alanine and Aspergillus fumigatus metabolism and biofilm formation. And so when I think about Aspergillus fumigatus, it's this saprophytic mold that grows in the environment, and really its association with humans is pretty recent. It's really only been associated with humans in the last 100 or so years with the advances of cancer chemotherapy treatment and different medical advances that predispose people to fungal infections. And so what I like to keep in the back of my mind at all times when I approach questions regarding this fungus is really that it evolved more or less independent of humans. And so its main niche in the environment is associated with decomposing organic matter, such as the classical example of a compost pile. And in this role, in this environment, it has an ecological role in carbon and nitrogen cycling. And really where humans come into play is when we come into contact with these spores. And so on this plate here, we see the green in the center, and that green is billions and billions and billions of these asexual spores or canidia. And they're very easily aerosolized. And so if you say take a walk in the forest and kick up some dirt or go do some gardening or something, you're likely to aerosolize these canidia, and then we can breathe them in. And in fact, most humans breathe in hundreds to thousands of these canidia every single day. However, for the vast majority of us, this is not at all an issue. We have really good mechanisms of clearance, both physical and immunological. And it's really only if one of those two barriers is disabled or suppressed in some kind of way that we become disposed to infection by this fungus. And so either a blockage in mucociliary clearance, such as what you see in cystic fibrosis patients, or a suppression of the immune or the innate immune system, such as is seen in cancer chemotherapy individuals or individuals that are undergoing transplantation. And so if one of these two mechanisms are blocked, the fungus is able to stick around long enough to actually germinate and start growing. And at this point, it becomes very difficult for the immune system to combat and fight this disease. And in individuals with innate immune suppression, so specifically, we end up with a large number of cases that in which this disease advances into an invasive form, and the fungus grows into these large biofilm structures. And ultimately, this all kind of gets to this larger question that myself and other members of the Kramer lab like to refer back to, and that's this idea of, what is a saprophyte doing in the lung environment, and how can we actually go about combating this infection? And again, thinking about the origins of this fungus and keeping that in the back of my mind, the fact that it's involved in carbon and nitrogen cycling really it screams metabolism as being a central feature to this fungus, and both in terms of metabolic capacity and that it can degrade and utilize a lot more organic substrates than, say, Candida albicans or Saccharomyces in these canonical yeasts, either industrial or pathogenic. And then, but then it also has to be very metabolically flexible to survive in these compost environments where there are vast changes in nutrient gradients and also in things like oxygen. And our lab had, or at the time that I joined, our lab had really been studying this idea of oxygen deprivation and adaptation to a low oxygen environment. And a big thing that really spurred this is that, again, we can not only see this in, say, a compost pile, but we can actually see a lack of oxygen at the site of infection. And so here's just a immunohistochemistry slide, or immunofluorescence slide of lungs of mice that have been infected with Aspergillus fumigatus, shown in green. And we see, using this hypoxy probe dye, these large regions of the biofilm that have less than approximately 1.3% oxygen, or in other words, are heavily oxygen deprived. And so that's really where everything was when I joined the lab, and what spurred a lot of my investigation into Aspergillus metabolism is this idea of how is it metabolically adapting to these low oxygen environments. And we had a number of, so I initially looked through a number of RNA-seq data sets, metabolomics data sets, and this really interesting theme kept on popping up. And that was this idea of alanine metabolism. There's a very strong signature in just about every data set that I looked at. And initially this was a little bit puzzling, since if we think about alanine metabolism, there really isn't any direct reducing potentials or ATP or otherwise chemicals that are involved in either redox balancing or energy generation. But then in looking beyond the fungal field, we really start to see that this alanine metabolism in association with low oxygen is a common theme across broad evolutionary distances. And so the two examples I'm featuring here are that if you waterlog plants, the roots start to produce massive quantities of alanine as the oxygen is decreased. And it's thought in the plant system to be used as a carbon and nitrogen storage molecule for when it becomes reoxygenated. As you can see, it very quickly depleted from the roots upon reoxygenation. But then also in humans, actually, we have this interesting association in our muscles, and in that if we exercise and our muscles become oxygen deprived, we normally think of glucose becoming or being fermented into lactate, and that is in fact very much happening. But the second most abundant metabolite to come out of that reaction is actually alanine, whereby it can be described in this interesting cycle called the Cahill cycle, where your muscles will produce alanine. It can be shuttled to the liver, where it can be regenerated back into glucose, and then the glucose can be shipped back to the muscles, thus completing the cycle. And so it's a carbon exchange mechanism between glycolytic and gluconeogenic organs. However, how this alanine metabolism might play into a fungal pathogen, or even really at a molecular or mechanistic level, really hadn't been explored. And so if we look at the gene that was actually popping out and showing up in all of these data sets, it was this gene that we're calling AlaA. And so it's an alanine aminotransferase that simply interconverts pyruvate and alanine using glutamate as an amino group donor. And so again, no reducing potentials involved, no ATP involved, but it does sit at this very interesting intersection between glycolysis and nitrogen metabolism. And thus we decided to try to investigate and see if the fungus was trying to tell us something with these transcriptomic and metabolomic data sets. This immunofungal genetics lab, naturally, I made a couple of knockouts of this strain in different, or of this gene in different backgrounds, and we can see that on minimal media that does not contain any amino acids, it is able to grow robustly, and thus it's able to at least make enough alanine to support this kind of growth in protein synthesis. And so deletion of this gene does not yield oxytrophy. However, in the initial test experiment, to see if it had a growth defect in 0.2% oxygen or a highly oxygen limiting environment, which would go along with our initial hypothesis, we found that there actually wasn't any substantial impact of this gene being deleted in an oxygen limiting environment. And furthermore, just to show that it is in fact actually an alanine aminotransferase and involved in alanine metabolism and not simply a misannotated gene or something that looks like an alanine aminotransferase, but it's say actually an aspartate or otherwise aminotransferase, is that if we grow it on alanine as a sole nitrogen source or a sole carbon source, as shown in the middle and the right, we see a substantial growth defect of the null strain. And so it is involved in alanine metabolism, but it does not seem to be the critical pathway that we were hoping to find for 0.2% oxygen adaptation. However, we went back to the drawing board and started to think about where Aspergillus might actually be encountering these in a more natural setting. And the thing that really came to our minds was in the context of a biofilm. And so Aspergillus fumigatus is able to form these very robust biofilms, both in vitro and in vivo. And as these biofilms develop and mature, Caitlin Kowalski in our lab was able to show that they they develop these very steep oxygen gradients with the base of the biofilm being near anoxic. And so I wanted to know if these more natural settings of oxygen deprivation and oxygen gradients, if in those contexts AlaA would have a role. And so thus I grew the fungus as a biofilm, and that's where stuff started to get really interesting. And so if we grow the AlaA null strain as a biofilm and then test for its ability to actually adhere to a surface, we see that it's very deficient in its ability to physically adhere to abiotic surfaces in this case, despite the wild type and the reconstituted strain being able to very robustly adhere. And this is where, again, things got interesting and started to lead us down into what this gene might be important for in Aspergillus fumigatus physiology. And the main reason I say that is that adherence had already been described pretty well by the Shepherd lab, and that the primary adherence factor for Aspergillus is this galactosaminogalactan polysaccharide, which is a heteropolysaccharide composed of galactose, N-acetylgalactosamine, and galactosamine. Interestingly, it's immunomodulatory. And again, if you disturb the generation of this polysaccharide, either in its synthesis or its maturation, you completely eliminate the ability of the fungus to adhere to abiotic surfaces. And so to look at this, we took a microscopy-based approach. And so we can image the first 300 microns of the biofilm using this Cockflor white dye that'll stain chitin, and thereby as a proxy for biomass, we can visualize all the hyphae. And you can see they form these really nice structured communities, as I showed earlier. However, we can also stain for the galactosaminogalactan polysaccharide, specifically using a FITC labeled soybean and gluten and lectin. And when we do that, we see a severe decrease in the staining patterns of the AlaA deletion strain relative to the wild type. And with the help of Ben Wicker and Carrie Nadell, we were able to actually quantify this and show not only qualitatively that it seems to be producing less GAG, but also that it is in fact making less of this polysaccharide. And additionally, we teamed up with the Shepherd lab to show that it not only is making less of this polysaccharide, but it also has a defect in the maturation. And so it's making less of it, and less of that is actually becoming or matured into its functional form, which would explain the adherence defect. So at this point, we can start to put together a model where we have this gene, AlaA, is involved in alanine metabolism, and then this somehow goes on to impact this extracellular matrix polysaccharide production and maturation, and thereby biofilm formation and adherence. And that's really fantastic and interesting, but I really wanted to know about other polysaccharides, specifically those of the cell wall. And so we went on to show that the cell wall structure is actually altered upon deletion of this AlaA gene. And that was fantastic, but I also wanted to know if that had any true functional impacts. And specifically, the example I'm going to show today is that of Echinocandin resistance. And so with caspofungin being the prime example. And This is particularly interesting for Aspergillus fumigatus since it is a drug that is very well tolerated in human patients. However, in Aspergillus, it tends to lead to these really weird phenomenon called paradoxical effects, whereby as you increase the drug concentration to a certain extent, it will continue inhibiting the fungus. However, if you increase the concentration even further, the fungus will start to recover growth and become less susceptible to the drug. And so you end up with this very interesting kind of U-shaped dose response curve, which you can imagine would be very problematic in the clinic, as if you don't hit that just more or less perfect dip in the U, you could end up having a very low efficacy of the drug. And so to do this, I grew up biofilms for 24 hours in our standard minimal media. We can treat with different concentrations of the caspofungin or echinocandins for 3 hours, and then assess metabolic activity using this XCT dye as a proxy for viability. And when we do that, we see that the wild type in blue it really doesn't care that much that the echinocandins are around or that caspofungin is around. It does very, very well in resisting this drug. However, in the AlaA null strain, we see is that the biofilms are essentially completely decimated by the presence of caspofungin. And then in addition, we see signs of the paradoxical effect in our AFT93 wild type biofilms that are eliminated upon deletion of this gene. And this also holds up for another echinocandin used in the clinic, mycofungin, where again, the wild type really doesn't care that this echinocandin is around. However, the null strain is completely inhibited by this drug's presence. And initially that was very confusing to me, since I thought that I had tested these strains for caspofungin sensitivity before and not seen anything. And in fact, I had. However, I did that using traditional methods that are used for drug screening, which is that you take the dormant canidial form of this organism and then apply the drug stress directly, either in a microtiter plate doing an MIC style assay or into an agar plate at a fixed concentration. And when we do that, we see that there's really no difference between the null strain and the reconstituted strain. And so we actually have found a really interesting example of a physiologically specific gene, in that it is required for biofilm resistance to echinocandins, as we see on the right. However, it is dispensable when the drug is applied at the canidial form. And so this is particularly interesting, since this gene would never have been found in a chemical genetics screen using these traditional methods that are very common for drug screening, as shown on the left, versus if you actually look in the physiological context that the fungus is going to be in during the infection process, which is that of a biofilm, as shown on the right. And so at this point, and this is really the summary of what we found in the MBio paper, is that we have this gene AlaA. We can genetically disrupt it or disrupt its function through chemical inhibition. And it's critical for alanine metabolism. And this then goes on to impact a wide variety of different phenotypes, specifically featured today polysaccharide regulation in GAG production and maturation, and cell wall alterations that ultimately impact biofilm formation, adherence, and interesting biofilm-specific echinocandin resistance. However, there's been this big black box that's been rather tricky to figure out for quite some time, and I'm finally starting to get some leads. And that is this question of what is the actual mechanism by which this alanine metabolism is going on to impact these polysaccharide regulation, or impact polysaccharide regulation, since there is no real direct or even indirect connection really to polysaccharide synthesis or polysaccharide precursor synthesis and alanine metabolism. And so in thinking about potential mechanisms, I really came up with these 3 top candidates. And one is that there could be a protein moonlighting function going on, whereby the protein has a regulatory function that is independent of its catalytic activity. And this is described for quite a number of glycolysis related enzymes in mammals and other systems. Or it could alternatively be really critical for metabolism and metabolic flux regulation. So the consumption or generation of alanine via this enzyme could be really crucial for balancing this metabolic flux, and that this could then feed back into, say, polysaccharide biosynthesis or polysaccharide precursor generation. Or finally, that alanine or that alanine itself could actually be acting as a signaling molecule, and thereby regulating polysaccharide synthesis and impacting the physiology of the fungus in that way. And in the MBio paper, we pretty sufficiently ruled out protein moonlighting using a catalytically dead allele of AlaA. And so that left these other two mechanisms that are rather difficult to entangle since they feed into each other. And you can imagine that if simply just adding alanine wouldn't necessarily just differentiate those two, but in doing exactly that, we had further suggestion that one of these two hypotheses is actually what was going on. And so if you supplement alanine into the culture and then test for adherence, you see not only a rescue of the AlaA null strains adherence, but also an increase in and further potentiation of the wild type's ability to adhere to surfaces. And so that, again, suggested that it was one of these two mechanisms going on. And I took a page from medicinal chemistry to try to disentangle these two. And so we can make chemical analogs of alanine whereby one of the three functional residues are mutated independently or in combination. And then this should essentially render it unable to be used for metabolism. However, if one of these is able to rescue the adherence or other phenotypes, then it would suggest that this is metabolite signaling rather than metabolism itself. And so I did exactly that and found that we can modify the carboxyl group in almost any way that I've tried so far, and more or less perfectly recapitulate simply the addition of alanine itself. And so these analogs are unable to be used in protein synthesis or alanine metabolism in any way, but they retain the ability to rescue this phenotype, thus suggesting that it is actually a metabolite signaling mechanism rather than through metabolism itself. But this still brings up this question of, what might alanine be signaling? And the one bit of evidence that we have, or one suggestion or hint, is really where it sits in metabolism, that it sits at the end of glycolysis as an offshoot of pyruvate. And so then in thinking about central carbon metabolism and the main regulatory factors that are described in that for fumigatus, it brought up this idea of carbon metabolite repression, which is essentially a way for the fungus to selectively eat its favorite food over other types of food. And so say if you really, really like steak and you have steak potatoes and say Jolly Ranchers on a plate in front of you, you're going to primarily eat the steak. And this is the case if you are completely repression locked, you will solely eat the steak and only the steak. However, in most strains, you're slightly metabolically flexible. And so you'll eat primarily the steak, but every once in a while take a scoop of potatoes or pop a Jolly Rancher. And then on the other end, we have de-repression locked, where you will just simply throw everything together in a bowl, mix it up perfectly into a smoothie and eat it. And we have mutants that fall along this spectrum, whereby if you have the wild type, that again is flexible, and so it eats primarily glucose in the case of Aspergillus, but every once in a while we'll take a bite of something else, such as ethanol or amino acids or other carbon sources. And then if we delete the canonical regulator of this carbon metabolite repression, CREA, we end up with a de-repression locked strain whereby it eats everything all the time, no matter what. And on the other hand, if we delete a repressor of CREA, this gene called SNF-A, we end up with a not a truly repression locked strain, but a hyper-repressed strain where it will more preferentially eat glucose over anything else than the wild type. And we can assay where something falls on the spectrum using its ability to resist alcohol or resist eating this toxic analog of ethanol called alcohol, whereby if the fungus eats it and metabolizes it, it kills the fungus. And so thus the CREA mutants in this example would be extremely susceptible to almost any level of alcohol, whereas the SNF-A should be resistant, as again, it primarily will eat the glucose in the media and not sample the alcohol as frequently. And so to assay this, I established biofilms in our typical glucose minimal media for 18 hours and then replaced with fresh, again, glucose containing media plus or minus the alcohol compound at different concentrations. And then we again went on to use metabolic activity via resazarine reduction as a readout for or a proxy for biomass and viability. And when we do that, we see in green here that the Kerkaert strain is completely, is extremely sensitive to this compound, whereas the SNF-A in light blue is more resistant relative to the wild type that is in dark blue. But what is really interesting and striking is that if we look at the AlaA null strain in yellow here, we see that it's actually even more resistant to ALA alcohol toxicity than the SNF-A mutant, suggesting that it is even more repressed than the SNF-A knockout strain that's the canonical repressor of Kerkaert. And this leads us to our working model, and what I'm trying to test right now is that if glucose is present, it will be undergoing, or it will be utilized through glycolysis at some rate. And some proportion of this is going to end up in alanine via this AlaA protein. And then the concentration of alanine would thus be is essentially a proxy of glycolytic rate within the fungus. And so the level of alanine is able to signal potentially through the carbon metabolite repression system, and then hypothesize that it acts as more or less a negative feedback loop to regulate glucose distribution. And so if glucose is abundant, you need to be able, it's very advantageous for you to utilize some of that for cell wall synthesis and extracellular matrix polysaccharide synthesis, since these are very both carbon and energetically costly molecules to make. And so thus it acts as kind of a balancing or a carbon balancing signal in this manner. And so then in a AlaA deletion strain where we delete its ability to actually make alanine via this pathway, we lose the ability to generate this signal, and thus the amount of carbon that you shuttle to the cell wall is decreased as the fungus essentially thinks that there is less of it available, and it prioritizes its use for energy generation. Then what this looks like in a biofilm is that in a wild type biofilm, you will have a high level of GAG production and a high level of deacetylated GAG, thus leading to robust adherence. And in addition, you have high cell wall integrity leading to echinocandin resistance. Whereas in the AlaA deletion strain, you have decreased GAG, decreased maturation of the GAG, and thus decreased adherence. And in addition, you have a lower cell wall integrity and thereby are echinocandin sensitive. And with that, I'd like to thank the lab and collaborators, Carrie Nadell, Ben Wicker, and Don Shepard and Francois were all critical for the GAG analysis. And additionally, my committee members and the Anne Levanway in our microscopy core is a fantastic resource. And with that, I can take any questions, or I guess we're moderating questions.
Yeah, thank you, Josh, for a beautiful talk. And first, so please enter any questions in the chat, but first I'll send it over to Liz with questions for Spyridoula.
Great, thanks. So Spyridoula, Jessica has started us off with a little bit of an off-the-wall question. Hopefully you've had some time to think about it, which is, do you think that this candidalysin molecule could be used in combination with EGFR inhibitors to improve anti-cancer treatments?
Yeah, so I see the point. So with the EGFR inhibitors, you block the EGFR-ERK pathways, while with candidalysin, you can increase the p38 axis, which can be tumor suppressive. The issue is that there is now growing research that p38 can also be promoting tumor, as it's promoting survival and resistance to stress. And in our case, you saw that p38 also activated an anti-apoptotic peptide. So I'm not sure how this could work. Something to try on, I guess.
Yeah. Okay, thanks a lot. So if anybody has any other questions that they would like to ask Spyridoula or Joss, please just drop them in the chat. There's no question that is too silly if you're a graduate student in the room and you just want to clarify something. Please also feel free to add that in. That's why we're here.
Yeah.
If I could ask a question of Josh, Jessica.
Of course.
Yeah, so I really enjoyed this part, looking at the difference between biofilm growth and just the normal MIC assays. You've hit exactly the right point, which is that we need to be considering the context in which these infections are growing. I didn't quite understand the connection between the why your model accounts for biofilm sensitivity, but not for growth, if that makes sense. Do you see where I am, where my confusion is?
I think so. So you're getting at the like why a Canidia would be sensitive versus a biofilm
Yes,
in
exactly.
different contexts.
Yeah, exactly.
And I think that essentially has to do with the physiology of the fungus when it first encounters that stress is going to impact how it responds to the stress. And so with a Canidia, before it's actively growing, it can yield, or it's able to likely rewire itself in a way that it yields, say, stress response A, versus if you already are committed to growing in a certain lifestyle and then have a stress that you encounter, you're likely to have a different stress response.
Yeah, I'm just wondering if there are pre-activated stress responses in Canidia that aren't present in growing biofilm. So for example, Canidia are like primed to detoxify reactive oxygen species or something like that.
Oh yeah, I would be very surprised if that was not the case, especially given some of the work coming out from like Michel Momani and other groups looking at how Canidia, essentially the environment that you grow them in, and ends up impacting what RNAs are actually present in the dormant Canidia, and so in what way they are primed for the next environment. And so yeah, I would guarantee you that if you took them from different environments, you'd end up with slightly different phenotypes depending on what those prepackaged mRNAs are and the exact structure of the Canidia.
Yeah, great. Thanks so much.
Right. I have another question for Josh, which is, I'm very curious about this idea that sort of gag is maybe a safety mechanism. If there's too much glucose around, the fungus can shuttle something into cell wall or polysaccharides. And so what do you think the dynamics look like in something like a cystic fibrosis lung or the compost pile? Do you think that there's sort of a period of breakdown that increases the local carbon source concentration? And then you get a burst of, say, gag production, or how do you think that works as the fungus is breaking things down?
Yeah, that's been something that I've been thinking about more and more recently, and I think I think you hit it exactly right in that as it's breaking it down, it's going to have more of these fermentable carbon sources around that are easily converted to polysaccharide precursors and also able to go through glycolysis. And so I think it's not necessarily bursts, but it's likely it likely is in kind of like waves as the carbon availability changes based on, again, waves of degradation versus biosynthesis, and just maintaining that right balance between the amount of degradative enzymes it produces and the amount of actual like glucose or glycnac or otherwise that's available for it to consume.
All right, we have a question from Amelia Barber for Josh. It says, great talk, a bit off the wall in terms of questions, but how important is adherence in the soil or the compost environment for the fungus?
Oh, that's a fun question. I have no idea, and I would love to know the answer to that. Since you'd imagine that if it's sitting in the middle of a compost pile, it's got a lot of stuff around it to keep it in place. It's not just going to, it's not like it's in a river or something where it can be actively flowed away. And so, yeah, I'd be very curious to understand that answer. But the gag also has a number of other potential functions, especially in its cationic form when it's matured. And so the Shepherd lab right now is actually has this really fun hypothesis where they think that it's a more or less a polysaccharide toxin. And so the positive charge is able to actually disrupt membranes of, say, amoeba and other microbes that might be attacking it. And so it could have this dual functional role of if you are in an environment where, say, flow is occurring, such as a place that gets a lot of rain or something like that, it'll help with adherence, but then it also might have a defense mechanism in that it could be killing, say, amoeba and other attacking microbes.
Very cool.
Yeah. So Spyridoula, I have a question for you about this in vivo work that you showed, and I'll just preface this by saying I'm not an immunologist, so this might be a very naive question. So you showed that in your mutant you have a defect in early clearance, right? You get this increase in CFUs, but then by day 4, both the mutant and the wild type have been cleared, right? And you emphasize that this is because this suggests that P38 is really important for that early clearance event, right? But the sort of message that we get a lot of times in infection models is that early events are really predictive of long-term outcome, right? So when you saw that data, were you surprised by the clearance of the infection on day 4? And what's your explanation for that?
So yeah, my hypothesis on how could P38 be involved in this, if you remember the live cell imaging video, is by inhibiting P38, EGFR remains on the plasma membrane. So I guess that this makes it more accessible for the fungus to use it to internalize itself, and therefore have increased number of fungal burdens. Because that's one method of facilitating EGFR internalization by P38. Others can be ALS3 or even FA2. So by the time the host can employ the other pathways. And don't forget also that these mice were immunocompetent. They were not immunocompromised. Okay. So we expect clearance.
And does that treatment of the P38 inhibitor wane over time? So eventually you're seeing P38 come back?
We did not test this because we administered the P38 inhibitor from 1 day before the infection and for every 12 hours. We were following a published protocol. So the days of the harvesting, we administered the P38 inhibitor, and 12 hours later, we euthanized the mice. So we didn't test for later time points without the inhibitor.
This is sort of a follow-up question to that. Do you see any patient cohorts with impaired P38 function, and are those patients more susceptible to candidiasis?
Not that I'm aware of. I haven't checked that, to be honest, but it's something we could follow.
All right, I think that's all the questions that are in the chat. So,
all right, thank you, everyone. And we will see you next month, and hopefully at the Mycotalks at the end of this month. But thanks to our speakers for 2 fantastic talks.
yeah, thanks so much, guys. We're wonderful.